Okay, right. So welcome to this session 2 on the role of connectivity in the structure and function of shallow lakes. We're looking forward to these contributions very much. So I'm just going to give a very short intro of a couple of minutes, and then I'm going to chair the first 5 talks, and Laurence is going to chair the second 5 talks. And we're all going to keep an eye on one another. And really, I think we wanted this to make this as much of an opportunity for the people who've submitted talks to kind of give a very compacted version of those talks to inform the discussion, and for us to kind of discuss those talks as fully as possible, and then have a kind of sum up as the end. So I really don't want to take up much of the time. So really, I guess the rationale behind this particular session comes from the kind of the thing that we all talk about. We all talk about connectivity, both ecologically and socially, and the sort of overriding importance of connectivity. But while we talk about it a lot in depth, we don't really understand properly that well about the overall importance of it. And perhaps for freshwater systems above anything else, connectivity is one of those kind of quintessential properties of freshwater systems, and it's perhaps one of those kind of topics that freshwater science above any other sort of science ought to be able to own, this kind of topic of connectivity. So it's really important that we kind of make some progress in this particular area. And we can really all think about connectivity basically as the sort of the mechanism by which anything from energy, materials, matter, contaminants, genes, propagules, biota, etc., all are kind of moving around a landscape. And while we kind of often have a sort of very individualistic view of water bodies or habitats, we know that all these particular water bodies and habitats co-occur in a landscape, and connectivity is influencing every single water body and every single type of biota to a greater or lesser degree. It may not happen all the time. It might affect some sites more than others, or parts of sites more than others. But basically, it's one of those things that's kind of just going on continuously. And we can have a kind of fairly static view of a landscape, but really kind of on top of that landscape, we've got this sort of overlayer of movement of water, movement of humans, movement of animal and bird vectors, which is transporting other organisms, energy, matter, etc., throughout that kind of landscape. So hopefully in this session we can make a little bit more progress and exchange with one another in understanding this issue and the role of connectivity in these systems. So first up, we had a nice talk by Yandis in Brazil, looking at, let me see if I can share, here we go, looking at the role of rainfall and hydrological exchange in plankton dispersal and habitat homogenization in dryland systems. So if Jansen, if you're there, if you'd like to pick this up and I can take you through this, or if you want to share your own screen with your own slides, that's fine, just let me know.
Let me try. Can I share my screen?
Yes, if you wish. Let me just see.
I will not present more slide again. So you will open to the question now, or will you present the other thoughts now?
Well, I have just 2 or 3 slides of yours taken directly from your talk, and I was hoping you would just talk over them, and then we can take some questions. I'll stop sharing and you can share if that would be easier.
Okay, so I will wait till you finish, and after that we open questions. Okay, sorry.
Okay, Janderson, are you sharing your screen?
Hello, hello everybody. Janderson, are you going to share a screen or not?
No, I'm not.
What? We cannot see you. Do you have a problem in your camera?
Okay, I will open up. Sorry.
Okay.
Pelo menos eu ouço, né?
Now?
Now we can see you. Thank you.
Right.
O homem não falou nada, não?
So I understood from Nigel that if you would like
Não, mas eu
to
achei muito.
make a short summary of the main highlights of your talk, is that correct, Nigel?
That's correct, yes.
And I would like to ask everybody who is not presenting or answering questions to keep their microphones off. Otherwise, it will be too noisy. Pablo Binder, please. Yeah. Okay. So you can say.
Thanks, Coca. Thanks, Professor Nigel and Professor Laurence Carvalho. The work that's submitted to Shadow Lake meeting, it was already published in the Aquatic Ecology journal, but we decided to present it here again because it's closely related to main talk of the Shadow Lakes meeting. Because we address the importance of local environmental and spatial variable and structured meta communities of phytoplankton and zooplankton. The hydrology is well known, it's an important process in aquatic ecology, but the importance of this process of hydrology on the local environmental and spatial variable is still lacking in dry lands, especially in tropical dry lands, like the study that I presented. So our hypothesis that the spatial variables is more important during dry season when the lakes are isolated from each other, at least hydrologically. So we, in fact, observed that during the dry season, environmental and spatial variable were important to structuring phytoplankton and zooplankton communities. But during rainy season, only environmental factors were important. But what surprised me is since that this is not due to hydrological connectivity. We compare lakes with hydrological connection with lake with no hydrological connection. And when we compare this, the hydrological process seems not to be very important, at least to structured plankton communities. Hydrological connectivity is important to homogenize limnological variables like temperature, nutrients, but seems not to be important to structuring phytoplankton and zooplankton communities. So I think this is the main results that I'd like to present. So if you have any questions or that I talk more about the work, I can present.
Okay, so Jansen, I've put up some slides from your talk because I think it might just be easier if we did it like that, so that other people can see them and they can react to them and to your comments. But that was a good summary. In this slide, you can see you can really see the basic concept that you were testing around environmental heterogeneity in dry season and wet season, and how this extends to affecting beta diversity of phytoplankton, zooplankton. And then, as you say, we have this issue where when you look within river basins and hydrologically connected and non-hydrologically connected sites, the pattern is not always as we expect it to be, which tends to suggest that hydrological connection may not be as important in these systems. I think if I understand correctly.
Yes, yes.
and then this is kind of your interpretation of these results, and really the key point is around the fact that in the rainy season we have this much greater connectedness of lakes. It may not be hydrological. I think you emphasize the possible role of birds or wind or these kind of things, but the ultimate effect is that basically niche processes become more important, whereas in the dry season, when our lakes are more isolated from one another, they are disconnected. Once this happens, then we have much greater potential for dispersal limitation, which you tested through your method. So this is a nice demonstration of the kind of competing potential of environmental filtering and neutral processes within the same system of lakes at different times of year, depending on the wet or dry season. Is that a fair summary?
Yes, and I think this the more important implication of this study is that with the future predicted to the semi-arid region and the others, that will become more dry and hotter. Niche and neutral process will become more important for structuring plankton communities in these areas. So I think this is the more important implication of this study.
Okay.
And in this, we will have a talk here that will show that role, the important role of birds in the dispersion of microorganisms. Yes.
Thank you. So do we have any questions for Janison, please? So if you want to use your microphones, Jorge.
Yes, I have a question.
Jorge.
Have you tried to analyze the data annually to see on an annual scale basis which one is more important?
We have two different seasons, the end of the dry season and the end of the rainy season. We have not monthly or weekly data, but we have conducted analysis. One important point: we have conditions in, or the studies have shown that several droughts can lead to homogenization. This is some studies in the Spain, the wetland from Spain. And the question is, we have, in this study that I presented, we have a normal drought and intensive rainfall season. My hypothesis is that the extreme events of drought or rainy season can lead to homogenization of communities. So the next steps, we have conducted our studies, and we presented here in the Shallow Lake meeting, is that even during extreme droughts, there's no homogenization. There's a higher biodiversity in these systems. And in this case, we have studied monthly during a year. So, a several drought during something monthly in each lake. Other study that we present. Other question?
Actually, I was going to ask you why you think your results kind of apply well with phytoplankton, but not with zooplankton.
It's a very interesting question, and it's really challenging to discover that. Our hypothesis that for zooplankton during dry season, another study conducted in the semi-arid region from Brazil showed that during dry season there are more resting stays in these lakes, which can contribute to this more dispersal of this kind of organisms. Although there was a lower diversity and the abundance of aquatic birds, but there was a higher diversity of zooplankton resting stays, and this may contribute to counterbalance the wet season with dry season. I think this is one hypothesis, but it's a speculation. It's a challenge to say what happened.
Okay, thank you, Janison. If we could move on next to Helen Bennion, who's going to talk to us. Who presented the talk on the kind of role of the effect of connectivity in our ability to restore a lost aquatic macrophyte species in lake systems. I don't know if, are you there, Helen?
I am here, Nigel.
Excellent.
I haven't got the sunglasses or the sombrero, I'm afraid.
Disappointing. I'd lend you it if I could. Here we go.
Yeah, thanks, Nigel. And I should just say also thanks to Laurence and Linda for hosting this session. And perhaps it's just fit to say, given that I'm the sort of next one up here, that this is one of a number of talks that stems from this NERC UK hydroscape project, which I'm guessing was the sort of inspiration behind this special session. So the work I presented was very much linked to this hydroscape project, but it was just one element of what is a much bigger project. And I know you're going to hear about some of the other work that's been carried out later in this session. So this was very much linked to a paleo-ecological strand of the project. And what we were trying to do was look at what species have been lost from a number of UK lakes, and then think about how, could they recolonize if water quality is restored? So sort of taking very much a sort of restoration theme. I don't know what else you've got lined up for me, Nigel. So yeah. So obviously this image was really just to depict the various sort of aspects of connectivity within a landscape and those that might be important for lake restoration. And this is an example from the Norfolk Broads. It is Hickling Broad, I believe. Sorry, Nigel, if you just go back to that previous one. And it was really just to sort of get us thinking really about the fact that connectivity enables obviously propagules to be transported through the system, and sort of think about that sort of role of, if our sites are isolated, where do those source propagules come from if our sites are no longer kind of connected, or not connected to those sources any longer, or sort of possibilities for dispersal. So they were the sort of the bigger questions that framed this. And I did have a slide that sort of talked through the methodology in a bit more detail. So hopefully, you might have seen that. But in short, it's using plant macrofossils in sediment cores. And we're dating back to about 1850, taking that as our sort of pre-impact baseline, and using those macrofossils to look at what species have been lost. So focusing very much on aquatic plant species here, and then using contemporary maps to then look at where populations of those species of interest preside today. So I think that next slide probably is the best one then, Nigel. So yeah, so I just took us through 3 contrasting examples from UK lakes in these different kind of hydroscape regions with different sort of characteristic landscapes and catchments. So I'll just very, very quickly sort of talk through this one just to give you a flavour really. So this was one of the species I showed. This is Alatany hydropiper. So this is a species that's now rather scarce in the UK. And what we had at the top there is the 1850s data. So these are the bottom slices of our sediment cores that have been dated to 1850. And there's data there for about 8 lakes. And what it shows is that this species was present at that time in 4 of the 8 sites in that hydroscape. But then if you move through to the other map, which is the current day, the sort of 2010s, when we took the cores to see the surface sediments, you can see that now there's only 2 green circles. So that basically means that Alatany hydropiper is only found in 2 of the 8 sites currently. So it's been lost from the 2 circled in red there. And one of those is Loch Lebo, which is the kind of one that I used to sort of showcase the method. But what's quite interesting then is if you actually look at the current distribution, this little area of Glasgow that these sites are located within is actually one of the sort of strongholds for this species in the UK. It's not actually very, common across the wider sort of, at a national scale. And then what's quite interesting, if you look at the little pink squares actually show where we've found Alatany hydropiper in current surveys linked to the hydroscape project as well. So you can see it is actually present, and it's present within close proximity, but Loch Lebo is isolated. So even though this species is present in that neighbouring site they're shown in green, it sort of just poses this question, how would it actually get back into Loch Lebo should the water quality eventually be right for it, if it has only 10 kilometres distance to travel, but that site isn't connected. So it was kind of looking at those sorts of issues, and I showed some contrasting examples where we have sites that are now sort of hundreds of kilometres away from their closest their closest populations. So I think that you had a concluding slide there, Nigel, just a sort of,
yeah, well, you had the same sort of principle for NIAS and Littorella, yeah.
That's right. So that was the second example where you can see that the little hydroscape shown in blue there in the current distribution map is where we're trying to kind of restore this species of NIAS that's been lost, and yet the current distribution, as you can see, is really way over now in the sort of west coast of Ireland and the Outer Hebrides. So you know, hundreds of kilometres away. And I'll skip through the third example, because I think that gets the message across. So yeah, so it was just taking those three examples and then really sort of demonstrating, I think, that the connectivity really does play a key role in you know, in our restoration and sort of decision making here. There are clear implications for restoring sites that are now spatially separated from the source populations of these species that we want to get back into our lakes. So so there was kind of, you know, putting some questions out there, really. Perhaps something that we could discuss is, you know, what do we then need to think about transplanting or introducing plants in some cases? Do we just have to accept that we can't always bring back what was there before? Do we actually have to accept that some of our assemblages, even following restoration, might be new ones, this sort of idea of novel assemblages? And do we, you know, we have to reconsider some of our targets in light of these kinds of findings, you know, not have species targets, but perhaps thinking much more in terms of structural targets or functional targets. So I'll leave it there. Thanks.
Great, thank you, Helen. Do we have any questions for Helen? I kind of like this particular topic. Oh, Jorge.
Hi, Jorge.
Hi, Helen. How are you?
I'm good, thanks.
I was wondering
I'm in Brazil, but I'm okay.
That's good.
How are you?
Good, thanks. Yeah. I was wondering if you have also considering not only connectivity in space but also in time, like resting stages, like if the conditions are right, maybe the seeds that are in the sediments might just repopulate the lake, something like has been shown in ponds in Norfolk.
Yes, I mean that's obviously pinned some hope on that the seed banks might actually you know, come good and enable some of these to recolonize in that way, and that's you know, that's certainly something that you know, we're hoping could be the case. I know Laurence has been working, I hope I'm not talking out of turn, Laurence, but I know Laurence has been looking at a project looking at NIAS and the possibility of bringing that back, and hoping that, you know, that could actually be brought back through the seed, you know, through the seed bank.
Yeah, and the connectivity does come into our discussions with conservation agencies. So again, we're like yourselves, we're looking at how near the nearest sites are. Do we try and establish populations from the nearest sites? And do we want to re-establish in isolated sites that won't be impacted by invasive species or pollution, or connected sites so that they'll spread? So we don't have definitive answers to those questions, but connectivity is definitely part of the discussion of, you know, of deciding where to potentially reintroduce some of these rare plants. There is a question from Andrea Bottolo saying, you talked about dispersion in space, but what about dispersion in time? That's quite similar.
Essentially the same question.
Okay. All right.
Okay. I think probably in the interest of time, if you don't mind, Helen, we should probably try to keep moving. Thanks, Helen. So next up, we've got Simon Turner, also from University College London, talking about legacy metal pollutants and the sort of role of lake connectivity in potentially transferring these legacy pollutants and what impact that might have for lake food chains. So Simon, are you on board?
Yes, on board with you, Nigel, don't worry.
Excellent.
So this is, I guess this is a non-ecological a talk I gave. So this is about the metal pollutants in these systems and the effect that connectivity has on those sort of metal pollutants, not just spatially, but also, as Jorge just said, over time. And then also how some of these metals also now getting into food chains. So if you go to the next slide, please, Nigel. Thank you. So there's this, so we're working on the hydroscape project that most people have probably seen already in this this seminar, but there's this idea, there's these 3 hypotheses we're sort of like working on with this work, but the idea that, you know, sort of it's quite a given that upstream water bodies sort of perhaps trap contaminants and prevent their transport downstream. And then we're looking at the effect of legacy contaminants, because what we are very interested in is a bit like CO2 in the atmosphere right now, but you can, no matter how much we stop pumping contaminants into aquatic environments, we are still dealing with a huge legacy from the Industrial Revolution and sometimes earlier. But, you know, we've got 20th century, 19th century, 20th century sediments packed full of contaminants, and they are still releasing those into aquatic systems. So this work was just trying to quantify that difference. So are we looking at modern contamination, or are we looking at legacy materials coming in? So we know that because of things like climate change and sort of introducing more, you know, erosion and soil elements coming into aquatic systems, that it's very likely that any sort of recovery is being confounded by this continual story, this reservoir of materials coming in of contaminated sediments. And then what we're also trying to work out is then whether or not we can quantify whether or not these contaminants are then getting into the food chains and then moving up into aquatic biota, which is obviously a problem for ecological, for ecosystems themselves. And let's have another slide then, Nigel. Thank you. So this is essentially the work we were doing. So this is two of the hydroscapes in the UK. The red and grey dotted graphs there from this connected lowland system that Helen just showed a picture of, this very open system comprising of channels and lakes and sort of offshoot water bodies. And what this graph shows is the top 3 sites are pretty much, they are isolated water bodies. They're isolated hydrologically, they're slightly up. So this is Norfolk, they're about 100 meters altitude, but they are effectively acting as repositories for atmospheric contaminants. And just for 1950 there is marked because that's just as a sort of a marker to see where everything is at the same time. But so these are receiving a lot of lead, and these are lead and mercury profiles, sorry. So the lead and mercury are coming from fossil fuel deposition. So there's a peak in about the 1970s. But if we look at the connected system there, at the bottom ones, what you have there is you have an enormous amount of mercury shown there, the 1950 peak of about 1,200 nanograms per gram of mercury, and that is associated with the atmospheric pollution, but also probably some boat traffic and sort of like paints and mercury-based paints. And so in this connected system, if you release obviously a contaminant like that in a connected system, then it works its way through the system rather than sort of being isolated. So in this sense, a nice open connected system is probably good, you know, it's better for the ecosystems, but not very good for contaminants because it tends to spread it around. The other graphs there on the top are from the Lake District, and what that is showing is this sequence of sediment cores of lead, and it's showing that the peak of lead in the upland site, which is the farthest left at about 200 meters in the uplands, was receiving quite early atmospheric lead. And what that seems to have been happening is that has been slowly migrating its way through these sort of chain of lakes down to the biggest lake at the bottom called Lake Windermere. And so you can sort of see this sort of maybe a pulse of material moving through over time. But it's quite tricky to sort of actually say whether or not, you know, upland lakes receive a lot more atmospheric material, but then so do lowland lakes as well. But lowland lakes also have a lot more contributing catchments as well. So you get much more of a homogenized sequence rather than just purely atmospheric and catchment based. So it's quite difficult to sort of separate these material flows coming into these chains of lakes. But here's the next one then. So what we have here, the last one there, is just in the end, what we're able to do is using the lead-210 method I outlined in the talk, is we can actually quantify how much legacy material is actually getting into the surface sediments. And from these hydroscapes here in the Lake District, Norfolk and Glasgow, in Glasgow and Norfolk, you're pretty much looking at 95% of total lead entering UK lake sediments is coming from legacy material. In the Lake District, it's fairly similar, but you do find some sites like this one shown there just underneath where it says Lake District is an upland site in one of the wettest spots in the country. So we think that is still receiving quite a lot of contemporary lead fallout just purely because of the amount of rain it receives. So that's a bit of an outlier, but we're looking at least 90%, 90 to 95% of old lead is now entering lakes. But the main problem we found with this study was it's quite easy, obviously, to measure lead in sediments and soils, but when it comes to fish, so looking at whether it's being uptaken into the ecosystem, it is incredibly hard to measure lead in fish. And so the values there you can see on the left are less than 1 microgram per gram dry weight of fish flesh. And we know that lead doesn't bioaccumulate like mercury. If I was showing the mercury results, it's spectacularly much higher because you can see all these food chain systems but accumulating. But with lead, we're struggling to actually measure enough lead in sort of like in these fish systems to make sense of it. How's that?
So anyone got any questions for Simon? I haven't seen any in the chat.
So my main take on this, I guess, Simon, is that there's really such a vast amount of metals potentially sitting in these sediments, legacy metals, almost regardless of where you are to some extent in terms of landscape, that whatever you do in terms of changing connectivity is potentially going to have some really significant downstream effects that we might not necessarily sort of immediately expect.
Yeah, I think that's right. I think that analogy of the CO2 in the atmosphere is quite good. It's like you've got a bath overflowing, and even if you reduce what's going in a little bit, it's still going to be overflowing for a long time. And it's just things like thinking about management, like, you know, opening up these systems. By opening up systems, you just release these materials, and they you know, they will work through eventually, but you would expect to see quite a significant input, I think, if you started managing lakes by opening up systems.
Thank you. Do we have any other one other quick question before we move on to Jorge?
Oh, that's a good question, Linda.
Okay, so as the pulse moves down the chain of lakes, why does it take 50 years to get from Grasmere, which is in the Lake District, to other lakes in no time at all?
Well, so in Grasmere, it takes 50 years to get to Grasmere, but the distances and Grasmere and I can't remember the other lake next to it. The lake next to it is maybe 100, 200 meters away with quite a significant river flowing towards it. So I think they probably received the same amount of sediment almost contemporaneously, and then it takes a little bit longer. And the last lake I just showed there was the southern basin of Windermere, and I think there's these intra-basin effects which have a very significant effect on the sediment profiles and sort of like the timing you have these things.
Okay, that's great. Thanks, Simon. If anyone's got any more chat for Simon, if you want to put it in the questions, if you want to put them in the chat. But can we move on to Jorge next, please?
Okay, yeah, this is what we're representing here is just the work that we have been doing over the last 15 years or so, trying to understand like how multiple stressors interact on a lake landscape to drive the aquatic biodiversity. If you pass to the other one. So we have been focusing on a lake system in Northern Ireland that, as you see, is formed by a large main lake, Central Lake, and connected to it, there's a lot of satellite lakes that vary in degree of connectivity and also in degree of eutrophication. And we've been trying to understand how all this, like eutrophication, connectivity, how they all interact over space, but also over time, which is something that tend to be missed on meta-community studies. If you pass to the other one, so we've been doing like monitoring surveys, but also we're taking sediment cores from the lakes and comparing what happens over space and time. And we have found that as in other meta-community studies, the environment plays a big role, especially in structuring macrophytes, but connectivity is a big important thing, which not only is driving the biodiversity, but also structuring the environmental variation. And in this sense, contrary to what we saw yesterday in the United States, it's more like we saw in the Brazilian example of today. Isolated sites seems to be suffering more for local process, like for eutrophication, while connected sites are more influenced by the connectedness. Diversity is greater in the connected sites, but also invasive species are more located in these connected areas than in the isolated sites. So it's quite complex. If you pass to the next slide, please. And other things that we have seen is not only in terms of diversity, presence, absence of species, but also in terms of variation in relative abundances. And what we have seen is that connected sites are more heterogeneous, more diverse, whereas isolated sites tend to be more homogeneous, likely through the effects of eutrophication. And in time, I think that's perhaps the most interesting result is that we found that pre-1950s, which was the time that we think eutrophication was less of an issue, the location in the landscape was the main driver in terms of differentiating lake communities. But once you get eutrophication into the history, lakes started to converge towards a kind of similar, more similar association, and each lake, depending on its degree of eutrophication, starting to respond independently to what was happening in the system. So we have like one response that is spatially, but also a different response that is temporarily. So it's if you pass to the next one, which I think is we can see in this diagram basically that especially we have the main lake, which at the moment is acting as a biodiversity hub, receiving a lot of propagules from the associated satellite lakes, more diverse, more opportunity for species to grow. On the other hand, we have eutrophication effects increasing in the isolated sites, but temporary in terms of time, eutrophication is also acting individually in each lake. So what we believe is that if this continues, eutrophication will slowly starting to spread across the whole landscape, and what we're going to have at the end is eutrophication dominating over connectivity, and maybe we're going to have like the same homogeneous assemblages of macrophytes and invertebrates across the whole landscape.
Okay.
Yep.
Thank you, Jorge.
Okay.
I have a question, but I want to give everyone else an opportunity. Let me see. Okay, so we have a question here. I'm curious if you have an idea of why you found that isolated sites are more eutrophic, whereas Kendra presented the reverse for the US in her presentation yesterday, which was one of the things I was going to ask you as well.
Yeah, I think in the USA example, the isolated lakes, from what I understand, they tend to be more like mountain isolated lakes. Here we're talking about the same landscape. So in this sense, isolated lakes are more likely to suffer from nutrient inputs from agriculture than for comparing to the mountain USA lakes. So I think it's a matter of scale. Maybe if we put this into a larger scale and we put upland lakes, we're going to have that isolated lakes are different.
Okay, and Robert Hatchnick is asking, do you think eutrophication is directly affecting connectivity, for example, through propagule pressure?
Oh, that's a really good question. Probably yes. In some ways, yes, especially from the isolated lakes into the main lake. I think it's bringing propagules or more dominant species. But I think it's because this system, we have also several mussels invasions that I forgot to say in the main lake. So it's not only eutrophication, it's also the effects of other factors that play together. So it's not only eutrophication.
Sure, I was about to mention about zebra mussels because obviously, you know, you have a system where, in a sense, where the fact that your zebra mussels have got to your most connected lakes is almost buffering the effect of eutrophication in those lakes by maintaining a clear water state. But maybe over time, do you think that there is a chance that the zebra mussels will eventually colonize the more isolated lakes, and that you know, you have this sort of transition, and then the isolated lakes might actually start to recover a little bit more?
Yes, it's probably. The thing is that the satellite lakes, the substrate is quite soft compared to the main lake. So the main lake is the sediments. It's still a rocky sediment lake. The other ones are more softly. So I don't know how well can the zebra mussels cope, even if they go to the other sides, if they will cope well enough to have an impact. But yeah,
just one quick last question from Annette Janssen. So well, I'll let Laurence ask a question of you another day. But she's asking, could you see differences between algae and macrophytes? I can imagine that algae will profit more immediately from connectivity.
We haven't done much in terms of algae, but what we have seen is that macrophytes are the ones that are driving the changes down the food web. So it's probably yeah.
Okay, and Linda had a final question about why the total nitrogen levels, are so low in this system for a temperate lake, but maybe you can pick that up with Linda another time, because I'm aware that Laurence's talk is next, and he'll never speak to me again if I don't give him enough time to talk about it. So Laurence, you're next up.
Thanks, Nigel. So this is part of the Hydroscape project as well, but rather than looking at sort of biota responses, we wanted to sort of try and look at shallow lake functioning. And so we were interested in looking at the phosphorus to chlorophyll ratio. And from that, we had to look at the individual responses to both TP and chlorophyll. Do you want to move on, Nige? So yeah, but we weren't just, again, we were interested in three sort of main types of connectivity. Hydrological connectivity associated with the catchment. You can see this with the dotted line highlighting the lake catchment there. And then there's landscape connectivity, which is highlighted by these sort of green and yellow bands emanating from the lake boundary of increasing buffer size. And then we were also interested in vector connectivity, and particularly in this study in people and fishing visits as a potential impact. And then we were looking at how stress affected these three responses as well. So the responses were, as I said, total phosphorus, chlorophyll, and then the ratio of chlorophyll to TP. So we were looking at how stress and connectivity interact. With the stress, we're really looking at percentage urban area in the catchment and percentage agricultural area. Sorry, either in the catchment or in the buffer zone. And so, for example, for total phosphorus, you can see here for shallow lakes that the percentage of urban land in the catchment had a significant positive impact on total phosphorus, as we sort of can imagine. Catchments with high slopes have a very negative effect in deep lakes. And we do see an interaction between the stressor, which is agricultural land, and potentially a vector of connectivity, fishing visits. affecting positively TP in deep lakes. So you can see quite a big difference between shallow lakes and deep lakes, and also in the percentage deviance explained, a lot more explained in the deep lakes. For chlorophyll, interestingly, we didn't see any significant results of our measures affecting chlorophyll in shallow lakes, apart from geographic location and whether it was a clear water lake or a brown humic lake. Whereas in deep lakes, we do see influence of agricultural land, fishing visits again, and also a measure of landscape connectivity, the amount of river length in the 2 kilometers surrounding the lake. So a sort of density of the river network. And so when we saw this result, we started to think that fishing visits actually probably wasn't a connectivity measure affecting chlorophyll directly, but may actually just represent the amount of fishing pressure. And so it may be that lakes with a lot of fishing visits have a lot of fish in them, and those are having negative effects on chlorophyll due to the impact on zooplanktivorous fish on zooplankton. And so then when we look at the ratio, which is what we were really interested in, looking at how it may affect system functioning connectivity, we basically see a combination of those two things. So for shallow lakes, the percentage urban area in the catchment is the only sort of thing that we really see significantly affecting the amount of the chlorophyll to total phosphorus ratio, and that's largely through its effect on total phosphorus. I think, I can't quite remember now. And then in deep lakes, we see the opposite. So we do see in deep lakes a lot more of an effect of connectivity, but it is generally connectivity in the local landscape that is really affecting the ratio in deep lakes. And it highlights the fact that, you know, what's happening in the local landscape very close to our lakes is a really important area to manage if we're trying to manage chlorophyll and total phosphorus. I think that's that. So yeah, to conclude, shallow lakes, deep lakes have very different controls. We get greater, clearer explanation in deep lakes. The shallow lakes are, as we know, much more complex. And I think the important thing is that local landscape connectivity, particularly pond count and river length, we found to be really important in deep lakes, and that's close to the lakes themselves. I'll leave it there, Nigel.
Great, thank you, Laurence. That was very informative in terms of addressing this, the interaction questions, which we are sort of particularly interested in around connectivity and stressors. Robert's just asking about whether fishing visits scale with stocking, and can you say a tiny little bit more about the sort of derivation of the fishing visits thing?
Yeah, so that was based on very intensive surveys of how people use the environment in, it was actually just covered England, that, so we were able to get measures of numbers of fishing visits for individual water bodies around England and create a model. This was work by Dan Chapman at the University of Stirling, but create a model for predicting numbers of fishing visits at lakes across England. And it is really, we think it's a measure, a proxy of how many fish are there, fish stocking, and therefore their sort of top-down control, rather than sort of transfer, people, coming and adding nutrients to the water body.
Yeah, and Linda's asking whether the fishing effect correlates with urban areas, although we do know that stocked lakes are not necessarily in urban areas.
Yeah, so there was no interaction. We did how we selected the variables was a sort of a forward selection process, because we had so many connectivity measures and stressors and typology factors. We first did a forward selection, a variable selection process with random forest, and so if fishing visits tends to always come out, and so it would, if it had a correlation with some of the other stuff, then that would maybe not come out in the selection process.
So there's a few additional questions there. One particularly around what's the potential role of pond numbers upstream in terms of the significance for deep lakes. I can't remember what the signal was there, whether it was TP or chlorophyll, I've forgotten, but I don't know if you can...
So yeah, this is a study, but we were looking right across a national picture. So we had about, I can't remember how many lakes we had in the end, also got 300 or 400 lakes across the whole of Great Britain. And sorry, I'm just forgot what was the question again?
Yeah, what's the significance of ponds upstream in the catchment for the signal in, I think it was, forgot which lake it was now.
Yeah, so I think what I was getting at is that this was really an empirical study looking at pattern, and I think it's actually just raised lots of questions in terms of, well, what are the processes going on, why are we seeing these patterns in our empirical study? So it certainly is giving us a lot of food for thought and where to look at next. But there are obviously roles for ponds in terms of nutrient uptake and nutrient retention. Is the main thing we're thinking of in that respect. They may again be a proxy for how impacted that catchment is around the lake.
Great, thanks, Laurence. So there's a few other questions there in the chat that might be worth a quick look over, but I think I should stop sharing now and let you share. Is that right?
Yeah, I think that's how it's going to work, Nigel. We hope. Right. So I'm going to be looking at the talks through the order that we've got in the spreadsheet, the session spreadsheet. So Paola first, then Amy, and on from there. So Paola, I don't know if you are available to speak through your slides. I've got 3 slides that I can talk through myself, but I'm very happy if you want to do that.
Hi everyone. Yes, I can make the presentation. First of all, thank you, Laurence and Needle, for moderating the session. Well, sorry for my English, but I'm trying to do my best. I'm going to make a quick introduction to this work, which is about the spatial and temporal variability of nutrients in response to alternative states. The study site is Laguna del Sauce, which is conform a system of 3 connected shallow lakes, El Sauce, Cisne, and Potrero. This system has a special importance because it is the second source of drinking water in the country. Despite on that, it is a eutrophic system, and the cyanobacterial bloom have become more intense in recent years. That increase habits relate to nutrient inputs from the basins as consequence of agriculture and cattle production intensification, and the significant growth of urbanization without waterway infrastructure. Most of the time, that blooms and domination of phytoplankton occur on the biggest system, El Sauce, while the other one, Cisne and Potrero, are covered by submerged vegetation. So we wonder if the spatial coexistence of alternative states is conditioned by nutrient gradients. To answer that, we analyzed the data of 5 summer seasons from different sites of the space to catch temporal and spatial heterogeneity of the system. For most of the season, we found a spatial variability for both nitrogen and phosphorus, total and dissolved, and also a significant difference between the different seasons for most points in the space. This variability can induce to this variability can induce to the alternative states on the composition of the primary producers. However, when we analyze the variation of chlorophyll, we found less variation and different gradients from the nutrients. that indicate that there are another drivers involved on the response of the blooms and the system configuration. So we also analyze the variation of other physicochemical properties, but we didn't find a unique driver forcing this variability. So we conclude on that there are many external and internal drivers working together, and the interaction between those are really dynamic on time on the space. So if you want to show the second slide.
That one, Paula?
Yes, there we can see that in the main body, the lagunal sauce is where there is a slight tendency to higher concentration of nutrients than the rest of the system. And this can be because of in that place the integrates that come from Basin of Sauce and Basin Pan de Azúcar. So there is a connectivity of the nutrients concentration.
Can I move and you just can summarize from this, or do you want me to?
Sorry?
I'll move to the summary slide for you. Is that okay?
Yes. So we found that there are different properties that contribute to these alternative states in the system. There are some physical properties that affect, and there are some chemical properties that also are affecting. So the water level combines with direction of the winds, alter the turbidity and the transparency of the system, and that combines with the concentration of the nutrients, affect the dynamic of phytoplankton and macrophytes. Something like that.
Okay, that's great. No, thanks, Paula. I think it's a really
Sorry
nice...
for my English.
That's absolutely brilliant. It's much better than my Portuguese. So just want
Maybe if there are some questions, can write on the chat and I answer there.
Yeah, so we can write and answer questions in the chat. I had a question on the relative importances of the water level versus the wind affecting the sediment resuspension. Do you have, is it largely the water level that drove things?
Yes, the water level is really important in that system, because when the water level is less, and this is combined with the wind, quickly the system transforms to a very turbid, turbidity grow very fast, and the change is really fast. So that's determinate that the phytoplankton can't grow and affect directly the configuration of the system.
Okay, now I think it's a really nice study where you see the gradient in alternative states in the system associated with all these changes. I can't see any questions in the chat. Does anyone want to just open their mic and ask a question?
That's okay, I can write on the chat to answer.
Ah, yes, there is. Linda says, there is less variation in your total nitrogen levels than in your TP levels across the lakes. Is this because TN is limiting rather than TP?
Can I explain? Okay, so I was wondering whether the nitrogen levels in your lakes, which are all very similar, are because the nitrogen is running out and preventing more growth in phytoplankton, in algae, and TP is all over the place because it's not linked. But maybe we discuss it offline.
I think that total nitrogen could be the limiting rather than phosphorus, but in case that cyanobacteria that can fix, I don't know, fix is not the word, sorry, but can take the nitrogen from the air is not the limitant for them. So in that case, I'm not sure about that.
Okay, thank you.
Okay, I think we'll move on to the next speaker to keep up with time. Thank you very much, Paola. And sorry, I can't get PowerPoint to show it in slideshow mode, but it's maybe good so the speakers can actually see what slides I've selected for them coming up. So Amy, do you want to talk through this, your 3 slides?
Yes, thank you, Laurence. Can you hear me okay?
Yep, all good.
Great. So my talk was on the habitat range and condition for dystrophic water bodies in England. And it was really split into 2 parts. The first was an assessment of the likely habitat for this lake type within England. And the second bit was to follow up to look at specific species that are associated with these dystrophic water bodies to understand how the lake type might have changed over time and what pressures might have driven that change. So dystrophic water bodies, we're not entirely sure exactly what their habitat range and condition is for these, and we have less information than for other lake types because they haven't really been included in monitoring schemes such as the Water Framework Directive, particularly. And therefore, we started this this research with the kind of question, you know, what's the importance of these dystrophic water bodies in the context of England? And previous predictions of the importance of these systems had been rather lacking in data and were kind of based on site-specific knowledge to try and constrain how much habitat there might be within England. And so we wanted to try and improve on these predictions. And to do so, we combined a number of data sets, including the UK Lakes Portal, which categorised water bodies greater than a hectare, peat soils locations as provided in the map from Natural England, and also species-specific data provided by the Botanical Society of Britain and Ireland. And then importantly, we also included water bodies less than a hectare through the incorporation of the OS master map data. And this is really important because we think that a lot of the dystrophic water body class will fall into this, this classification of being less than a hectare in size. And so we incorporated all of these data sets. And within the species-specific data sets, we drew up a list of key dystrophic indicator species. And then we combined these with a list of generalist peatland species to create a presence-absence prediction across England. And in doing so, and in incorporating these data sets, we predicted an increase in the area of dystrophic habitat for England from about 12 kilometres squared that was assessed in the previous 2019 JNCC assessment to just over 24 kilometres squared. So we think there's more of this habitat out there than previously was estimated. And importantly, within our analysis, we found that water bodies less than a hectare were really important in contributing to the overall coverage of these systems within England. There are a number of uncertainties associated with our technique, and it may well be that we have misclassified some oligotrophic lakes that sit within peatland areas as dystrophic. And indeed, because we used a cutoff of the peat soil area where we required that at least 75% of the catchment needed to be peat for us to classify a water body that sat within it as dystrophic, there may well be some peat pockets that are supporting dystrophic water bodies that haven't been captured by our approach. So there's a real need for kind of verification of what we found, but we think that overall our data are suggestive that this habitat type is more important than previously suggested. So it was an interesting study in that first instance, and one of the things that we looked at was the dystrophic species, as I mentioned previously. And for the kind of clustered analysis, we found that there looked to be quite a lot of areas of losses of dystrophic species across England, and they were inherently associated with areas of peat degradation and loss as well. And so for the second part of this analysis, we looked at individual species and what had happened to these over time within England. And we looked at time periods pre-1950, from 1950 to 1990, and then post-1990, to try to understand how these specific species that can be found within dystrophic water bodies have been lost over time. And it's an approach similar to the data presented by Helen earlier on in this session. And it was quite nice to see the kind of the similarities between that study and our own, in that we tended to see that at the hectad scale, which is the 10-kilometre scale, which we used for our species data analysis, it seemed that individual hectads, or the peripheries of hectad clusters, as can be seen in these two contrasting maps here, seemed to be at greatest risk of loss over time. And although we didn't undertake specific cluster analysis, this seemed to be quite robust, particularly for Utricularia, which is shown in these two maps, which is bladderwort. And this species particularly was lost over time, and we found the same for other species which are strongly associated with shallow water bodies that could be found, for example, in peat pool type systems. And it seemed that these had been eroded from the landscape over time particularly. And that this kind of connectivity question meant that those that were geographically isolated perhaps were at greatest risk of being lost. And we know that there's also a lot of momentum at the moment towards restoring peatland systems in England and across Europe more widely. And I don't think that water body restoration is necessarily set out as an aim of a lot of these restoration schemes. And I think it definitely should be. And when thinking about how to restore these species, this question of connectivity in restoration schemes should be actively considered, as it seems that it would be important, and from these kind of findings that we have here. So that's just a quick run through. We also presented these data for other species, I should say, that showed increases over time. So it really is quite species specific, but if you think about the habitat niches and preferences for these individual species, this type of analysis can yield some really meaningful insights. So thanks very much, and happy to take any questions.
Thanks so much, Amy. As Amy pointed out, in all these talks, there's a lot more actually in the full talks than we've been able to show in the selected slides. So I do apologise to the speakers that I may not have selected the perfect picture, but just to point out something that in Amy's talk, if you go to the rest of it, you see that species with a wider ecological tolerances and a wider ecological niche tend to see this increase, whereas it's particularly very specialist, narrow tolerance species, it looks like there's been a decline. So I think that's really nice fitting with this idea of it's a combination of, you know, environmental tolerances and dispersal capacities and connectivity that will all come together to determine communities. So I think we've got some comments, certainly. An offer from Helen Bennion. She wants to get together, Amy, and look at utricularia loss through the paleo records and tie them in with your spatial analysis. I think it's really neat.
Yeah, I think that the BSBI data are great, but obviously the further back in time you go, the less and less observations you have to work with. So we kind of introduced this cutoff of pre-1950 just as a function of the fact that you have way less recording efforts if you look pre-1950. So we kind of aggregated that in itself. But yeah, I think the paleo question could be really nice to kind of get around that limitation in terms of records.
Yeah, I was just thinking it'd be nice to just extend that time scale, wouldn't it? I think utricularia, Jorge may correct me if I'm wrong, but I think it does actually preserve quite well in the macrofossil record, doesn't it, Jorge?
No, that's the thing. It doesn't preserve
well. Oh, does it not?
Yeah.
Not at all?
Not that I have. I think Tom at some point found some spines, but I haven't been able to find any spines of utricularia or seeds.
Okay, well, we abandoned that research project then, Amy.
It was nice while it lasted.
Maybe you could look at these key species and discuss which ones have the best remains, that you could explore that question with still.
Yeah. I have a question.
Go for it, Jorge.
And it's related with, you know, with acidification recovery. We're starting to see a lot of that. A lot of the sites become more DOC rich, and I don't know if that's going to lead to more dystrophic, at least temporarily dystrophic sites. Are you expecting to see this in the maps, like some recovery because of acidification recovery?
I suppose that they might transition from a kind of a clearer water to a more coloured water, but I don't know if that necessarily would couple with a species response if the nutrient dynamic isn't actively considered as well. I guess that one pressure on dystrophic species habitats is kind of nutrient pollution. So I think it would have to be kind of an assessment of colour as well as nutrient loading to see whether it could actually sustain a viable dystrophic habitat. But yeah, it's an interesting question, and we are seeing this increase in colour in a lot of systems at the moment that that could be a means of expanding the habitat range for this type, but I'm not entirely sure.
Okay, I think we need to move on, but there's some comments you can always answer in the chat, Amy.
Thanks, Laurence.
I'm going to move over to Riwan, who's going to present his PhD research.
Hello, everyone. So my first chapter of my PhD was about the within lake variation in zooplankton in a lake which exhibits a rupture of connectivity during the summer in terms of predation pressure, and we wanted to see how it will affect the relationship in the trophic network, especially the zooplankton compartment. So here you can see the fish spatial distribution in summer. So you have to see that in this lake we have only the brook trouts, which eat zoobenthos and zooplankton, and the yellow lines show the movements of the trout, of one individual trout during all the summer. And it's the same pattern for every trout we followed, so like 30 individuals. And in the bottom left, you can see the topography of our lake, with the left area, which is a shallow basin with less than 2 meters depth, and the right area with a deep basin where the fishes are. The idea was to monitor the zooplankton in the both basin to see how the community could shift in terms of composition and size spectrum between the two basins. So yeah, this is the normalized size spectrum theoretical framework. So it's a spectrum where we sum the biovolume in each size classes, and we normalize this biovolume to have a straight line in a log-log plot. And this straight line is well defined in the literature, with a slope as a proxy of trophic transfer efficiencies and the relationship between the predators and prey, and the intercept of the NBSS, which is related to the productivity and the total biomass of our system. So we will compare the intercept and the slope of the NDSS of our zooplankton population in the left and in the right basin. So here are the results summarized. And you can see on this graph the relationship of the intercept and slope for the week. So you don't have here the effect of the left and right basins, but I can tell you that when we have no fish, you have a much higher intercept, a much higher biomass of zooplankton with no predation. And in terms of slope of the in the SS, we have a steeper slope in the shallow basin without fish, which means that the small zooplankton is a lot more present in the shallow basin. And here are the variations during the season from the week 1, so July, to the week 11. It's soon October. So you can see that the intercept will go down during the summer with the fish eating zooplankton, and an increase, a light increase in the late summer and soon autumn, which will can be related to the newly accessible basin for the fish, because the temperature of this shallow basin goes under the lethal temperature for the brook trout. So this trout can go to the shallow basin and eat on other prey than zooplankton, for example, the zoobanthos. And this interpretation is really strengthened with the relation of the slope according to the week. You can see that the slope in the first weeks of the summer until late August are quite stable, and there is a drop of the slope which become less negatives between late August and September in 2 or 3 weeks, and the slope reach a new plateau after. And when we plot the temperature instead of the week, we can see that the drop of the slope is exactly is happening exactly during the weeks when the lethal temperature of the brook trout is when the temperature of the lake goes below the lethal temperature of the brook trout, when the temperature of the epilimnion, so the shallow basin is newly accessible to the trout. So we show that, yeah, so we show that the shallow basin is another ecosystem in our lake, and it's more than a refugee, because the zooplankton and the zoobanthos can thrive in this environment without fish predation. And when the thermal barrier is off during the October, we have a shift in our trout regime, diet. And we can ask ourselves, what if with the global warming, the rupture of connectivity in 2 areas of our lake can go longer and longer, and it can affect the support capacity of ecosystem and the accessibility to resource for the fish. So if you have any questions, I would be glad to answer it.
Thanks very much, Ruan. Any questions? A question from Nigel asking whether a change in water level as well as temperature can also affect the connectivity of the basins.
We monitor the water level during the summer, but I think it will not affect the connectivity, because the temperature alone is sufficient to impede the trout to go to the shallow basin. And between the 2 basins, the minimum depth is 1 meter. And we don't have so much fluctuation of our water level in our lake during the summer. So I don't think it's really significant.
And a question I do want to get onto, because I noticed Beta submitted this earlier in the chat, in the questions thing. Beta was asking whether you plan to look phytoplankton communities with us. If there are samples there, it sounds like she would be very keen to collaborate and extend your analysis.
Yeah, we sampled zoobenthos too to have another compartment of our traffic network. But about the phytoplankton, we use a fluoroprobe to monitor the fluorescence of our lake, and I think I will focus on that for the next chapter of my PhD. But due to some field limitation and fluoroprobe bugs, I will have to fix the data, but I think we will be able to see if there is a differences in the two basins in terms of phytoplankton, even if we don't have, I think we don't have enough phytoplankton sampling in the shallow basin.
Okay, all right, thank you. We should move on now, I think. So, sorry, we're a bit pushed for time. Thanks very much. And again, have a look at Laurence's talk. Some really great stuff there and some great graphics as well. So thank you. I'm going to move over to an old friend of mine, Robert Tachnik in Austria, who's going to talk about lake centrality. Now, Robert, I might have to hide a couple of slides just to move on. I've got them hidden, so be a bit wary of what I've got on there.
I think I'll talk about only 3 of the slides. Thanks, Laurence. Yeah, so this is an analysis on how lake connectivity may affect diversity of plankton communities in alpine lakes. It's a combined effort, the sampling from Wassercluster Lunds, where especially Christian Breiler and myself were out for sampling, and EAWAC, where most of the work was done by Blake Matthews, together with Julia Birtl. And I think we can go to the 3rd slide on your list. So for quantifying connectivity, the work that was actually done by David Cuniera-Moncosi, we quantified centrality according to an approach published by Bortogaray et al. In 2015. I think actually Anna was before in the audience. She might explain detailed questions on that as well. And we did calculate this centrality for a number of truncation distances. So the largest distance, 600 kilometers, considers a lot of lakes around the Alps for the centrality of our focal sampling lakes. And then we had a number of distances below that. The shortest distance was 6 kilometers, which is just considering the very neighboring lakes for calculating this centrality. And the next slide, please. We exemplify the results now with 3 diversity metrics. The first is alpha diversity or taxa richness, and we always do this for 4 different taxonomic groups: for zooplankton, phytoplankton, both analyzed by microscopy, and then 2 molecular groups, 18S protists, or actually more precisely, this is simply 18S, so that includes also some zooplankton, of course. And then 16S data, which is basically bacteria. And for all the 4 groups, we have alpha diversity, taxon richness. We have LCBD, the local contribution to beta diversity. That is how unique a given local community is relative to the network. And then the last parameter, the match with the environment, which in the next slide will be called environmental tracking. That's based on a CCA analysis. If you make a CCA for the full data set, we can check how good a given local site fits to the actual environment within the total analysis. And this we use as a parameter for environmental tracking. And in the next slide, please go to the next. Yeah, now you see for all the different distances, the correlation that we find for the 3 different diversity indices. And if you look on the right graph, the environmental tracking, for example, the blue and the red dots, they give you the correlation that we found, in this case for environmental tracking. Blue means for the zooplankton, between zooplankton environmental tracking and the centrality. So a positive value means that for this parameter environmental tracking and for this distance class, 6 or 60 kilometers, we found a positive correlation between lake centrality and how well the community was fitting into this environment. And this was quite consistent, especially for the short distance classes. And it was especially strong for phytoplankton, for zooplankton, and for the 18S data, which we should acknowledge also contains some zooplankton data. But it was consistent that it was positive for all 4 groups, even though not significant for 18S and 16S. And conversely, we found that for LCBD, log contribution to beta diversity, we found the opposite trend, that we found also more consistent signals for shorter distances, but now negative correlations. So meaning that the less connected sites were, the more unique their species composition was, the more especially they stood out in terms of beta diversity in the data set. And I come to the conclusion. Next, yeah. So for all groups, we found overall the strongest signal for the zooplankton, which matches that those were the largest organisms in our group, and we can understand that we find especially signal related to spatial processes in zooplankton. It was quite intriguing to see that the relationships overall were more consistent towards shorter network distances. That's an interesting result for us. It shows that we find effects of connectivity, not considering the largest distances, but rather like some tens of kilometers in that range actually. And yeah, overall, the environmental tracking correlates with centrality in zooplankton and 18S. That makes sense because 18S contains some zooplankton data. So in some follow-up analysis, we want to separate the 18S into different functional groups. Yeah, and the last conclusion is a bit really open to discussion, that this negative relationship between tracking and LCBD, especially in zooplankton and phytoplankton, that we see, it suggests that the well-adapted communities would not host that many exotic species, or you could also reverse it that the exotic communities would be less adapted, which is a bit provocative. And we really think that we have to do more on this type of analysis. And I think there's also quite some interesting connection to some of the talks that were in the morning from the that related to how unique or general environments are in how that fitting with the community we find on the landscape scale. I think with that I would be ready to take questions, but I think the time is very limited.
Thank you so much, Robert. Just in the interest of times, I hope you don't mind, Robert, but I think we should let Alan present, and then if everyone is happy to stay on, they can stay on for 5 minutes questions and to discuss both talks at the end. But I would just like that opportunity to Alan.
Hi, Laurence. Can you hear me okay?
Great.
Okay, good. I have terrible internet, so please bear with me. So I'm also part of the Hydroscape projects, and I think I take a little bit from all of the projects beforehand, and we're looking at both quantity, quality, and connectivity, and really thinking about the applied and conservation aspects relating to freshwater biodiversity. We've always considered quality of freshwater as a driver of freshwater biodiversity, but it's rare that we actually consider connectivity, although the speakers in this session will say otherwise. But actually comparing them in different landscapes and different species groups to see if we can find similar patterns is really interesting. Go to the next slide, please. So this is the central premise of the project, and if you're on the left-hand side of that plot, connected water bodies generally have a greater biodiversity. They have that ecosystem resilience, the stepping stones between habitats that can create more niches and more spaces for species. But what we hypothesize is the more stressed your environment becomes, that connectivity can actually become a burden. And we saw that somewhat in Jorge's presentation earlier on, but it's something we went on to study in this as well. So actually isolated water bodies in your landscape can possibly be a good thing if your landscape is very stressed. Next slide, please. So this is similar to what you saw before, and it's the 3 hydroscapes we're working in. They're highly contrasting in terms of their land use, but all of them are similar in that they have plenty of lakes and ponds and rivers, and shallow lakes too. And in this talk, I'll be talking just about the lakes and ponds. And within these, I've looked at the water chemistry, aquatic plants, moths, beetles, and dragonflies. So plenty of variation in there. And I wanted to look at the drivers of the richness in the, first of all. So these plots here, if it's on the right-hand side in blue, it's a positive effect. And on the left-hand side, it's a negative effect. And significant stars are on there. And there was lots of variable selection, but I tried to pick out the most important ones that are split across water chemistry, land use, connectivity, or physical aspects. And what we can see for the passive dispersing species, such as the macrophytes and mollusks, is that actually all of these things, whether it's a part of chemistry, a part of the connectivity, are having an effect on their richness, which isn't overly surprising. But what is actually the effect of water body area starts to be discounted. So ponds are almost no different to lakes in a way, because there's other drivers that are having a greater effect. And the variance is actually okay for these species, well, in an ecological context. Next slide, please, Laurence. The variance isn't quite as high for the active dispersers, perhaps not surprisingly. So the water chemistry doesn't really explain a huge amount of their richness. They're really affected by some of the sort of physical connectivity, such as lakes and ponds at higher altitudes have much lower species richness of beetles and dragonflies. Next slide, please, Laurence. So the one thing we wanted to look at was this interaction between stress and connectivity. So I have grouped the invasive non-native species here by pond and lake, just to really show that the lakes are always having more non-native species in them. And what we found is that lakes were much more connected in our environments as well. So on the right-hand plot there, you can see that the larger water bodies have a much greater richness of invasive species. And if you're in an urban environment as well, you're probably going to have lots of invasive species. The final thing to show there is the isolation, and that if you're an isolated water body, you have less invasive species. So not surprising, but this was the same trend in all 3 of the hydroscapes that we worked in. So final slide, please. So we did find that both or all quantity, quality, and connectivity does affect freshwater biodiversity, but this does depend on species group. There's a much less, a lower effect of the environment you're in. So we found that sedentary species such as macrophytes and mollusks are highly influenced by site quality, but there is an effect of connectivity. And we have been doing a bit more analysis on these. We still have some to do on the beta diversity, but there is a strong link that stressed environments are going to have more invasive species. So that should have implications for whether you're going to reconnect these areas or build more ponds, because it could spread invasive species, especially in urban large water bodies. I think that's it.
That's great, Alan. So what I want to do is open the floor, particularly to questions from the last 2 speakers. We were hoping to have a bit more discussion time, but we will have to, tomorrow, I should say tomorrow, we've got another 5 talks that Linda will be chairing. And because we've got 5 talks in an hour, hopefully we will be able to have a bit more time for general discussion of connectivity issues in general. The session is officially finished, but I'm happy to stay. I think if we stay for 5 minutes or however long people want to ask questions, but we understand if you want to move on and leave this session and have a break. So has anyone got any questions for Robert or Alan? I did have a question for Robert whilst if anyone's come up, and that's, I'm going to go back to his slides here. And I saw the closeness of the zooplankton and S18 data here, but they appeared to be separated, the richness and the beta diversity. Is there an explanation for that?
What do you mean by separated?
Well, in there, the response is the same for zooplankton and S18. They show a similar response. But here, there's a very strong response of zooplankton, but not the S18.
I don't know, but it may be methodological. That, I mean, the S18 methodologically comes from a very different pipeline. Yeah. So this molecular analysis finds much more rare taxa, for example, than does the zooplankton. So I can only guess that it has to do with this methodological difference.
I'm presuming the S18 includes phytoplankton as well, apart from cyanide.
Yes, actually it includes any eukaryote. Yes. Yeah. So and I just mentioned that it's not strictly only protists. I mean, we also get, for example, signal from the Daphnia. We know this from other experimental work, analysis, where we find probably from the Exuvia from Daphnia, we find in the S18 data also the signal of the Daphnia. So we assume that without filtering that there's also the rotifers and such data in it.
Okay, you could potentially analyze the S18 data in particular groups in that.
That's the plan. There is, however, a little caveat because we used a very general marker. That means our resolution on the different taxonomic groups is limited.
Okay, right, we've got a couple of questions for you, I think both for Robert. First from Esteban. He finds it really interesting, your results, the positive correlation between environmental fit and centrality. He expects the opposite pattern as more connected lakes experience higher mass effect and could present viable populations that are not well adapted to local conditions.
Yeah. It's a good point. But we should emphasize we are talking here not about lakes that are physically connected to each other. We really talk about discrete lakes. Here the connectivity is mostly through what you would call dispersal over longer distances, like with water birds or also with wind, but not by running water. So a high centrality means, okay, there are more lakes nearby, and the likelihood that a water bird is transporting propagules from one lake to the other is higher than for distant lakes. However, we are far away from talking of any mass effects if a water bird is transporting eggs from one lake to the other.
Okay, one final question from Dianica, who's one of our PhD student speakers tomorrow in the session. So we're looking forward to her talk. And she wants to ask Alan if that graph at the start on connected versus isolated in stressed conditions, whether that's theoretical or supported by data. Is it a hypothesis or a result?
It is both. And I'm going to try and show you my screen. I'll flip the camera.
Do you want me to stop sharing, Alan?
Yeah, that'd be great, actually. So, sorry, I only did this a few days ago. That's why it's not in the presentation. So it's not quite there, but you can see the stressor on the x-axis, the total nitrogen, macrophyte richness, and each of those points is scaled by the number of invasive species. So the connected sites at the top always have more invasive species. And as we move along to the right-hand side, there starts to be an interaction, but not quite there. So perhaps we've not got quite stressed enough sites, but it's still a work in progress. So I do wish I had this in my presentation, but I only did it a few days ago. So we're getting there.
This is research in action. This is.
I know.
So I think, Danica, there are results coming out of the project to answer that hypothesis, and we are seeing that certainly for invasive species, it seems to be true.
Yep.
Okay, anyone else got any other questions?
I had one, but I think it's related with what you show, because it was really interesting to see greater diversity in urban areas than macrophyte diversity than in the isolated sites. Is that just because of invasive species? For Alan, sorry.
There was a higher invasive species, and so this was just invasive species in urban areas. So basically, they had a greater diversity. For the plot that I just showed, separated invasive species and macrophyte and native macrophytes as well.
Oh, the one you've shown on your computer.
Yeah.
Okay. I think we probably should sum up now. Well, all I was going to sum up with was to thank all the speakers today. I think we have covered a real range of topics in terms of connectivity, and it just highlights the number of questions we still have, and that it is and from the two plenaries this morning, I think there's such a great area for further research for the freshwater community. And as Nigel pointed out at the start, in a way, we are lucky in that we can talk about isolated and connected systems a bit more explicitly in our freshwater environment. So I think it is a great area for continued discussion and continued collaboration. So just once again, thanks everyone for coming to watch the videos. Do go and make sure you watch the whole, all the talks in their entirety. They've got a lot of interesting results. And we look forward to seeing you tomorrow in the session that Linda will be chairing tomorrow, whatever time zone you're in. So that's all I'll say. Okay, thanks very much. And we'll see you another time tomorrow.
Thank you. Bye.
Thanks. See you tomorrow. Bye.
Thanks. Bye.
Thank you. Bye-bye.